This is a production of Cornell University.
I wanted to thank you all so much for the invitation. This is a real honor for me. And I wish that it was in person, but with the COVID restrictions and everything like that, we couldn't make it due to USDA restrictions. But and I'm a little bit under the weather today. I have a cold. I don't have COVID, but I'm working from home, and I hope I can deliver this and my voice doesn't give out. But again, my title, my slides are going to just be talking about domestication, breeding, genetics in Cranberry, and just a real honor to be here. And I hope next time is in person. So my slides are a little bit slow. I don't know why, but hopefully we can get through them. I am part of the USDA. So some of you know, I'm a research geneticist. I'm stationed at the University of Wisconsin-Madison, and we have 3 units. I belong to the vegetable crop research unit, but we also have the cereal crops research units, and we also have the USDA Forage Research Center. And the VCRU, we work in multiple crops, including carrot and garlic and onions and cucurbits, potatoes, and of course, cranberries. We are again embedded in the Department of Horticulture, where we function, as somewhat as a faculty. We help train students. We give some seminars. We may help with classes here and there, and we of course do the research. And this is an older picture of the department, but maybe some of you recognize some of the people here. Some of them are gone. Some of them are still here. The picture is more intended to show the number of faculty in the department, and then the number of scientists that are part of the USDA in the arrows. We're about a half of the department in terms of numbers. We have a few that retired this year, but we're hoping to replace them. So Mike Havey, Spooner, and Jansky, they all retired this year, or maybe last year, the end of the year. So we're hoping to recruit some new people. And but this is a really nice department to be in, and we have a strong presence of the USDA scientists. We also, my lab is called the Cranberry Genetics and Genomics Lab, and this is a picture of a Zoom meeting that we had not too long ago, where we couldn't still meet in person. So we've been conducting a lot of our meetings and efforts online and in person whenever possible. I typically have help train 4 to 6 graduate students, and I have a number of undergrads that help those grad students. And lately I haven't had a postdoc, but we have had many postdocs come through the lab, interns and invited scientists that come to our lab. And so it's always, this is probably one of my favorite parts of this job, is to be able to interact with students. And here's my outline. I'm not gonna go through it, but I have a lot of slides. A lot of them are very quick, just to show you like a picture, a visual of what I'm trying to tell you. So here's the background. Cranberries are a part of the Ericaceae family, where we have about 100 genera, 3,000 species. These are plants that are, although they like to be and they can survive underwater, they're not actually aquatic plants. They like actually well-drained acidic soils. And species that are related to cranberry that are interesting to me, because I work in cranberry, are lingonberry and blueberry. More recently, I had a trip to Hawaii, and I encountered a vaccinium called Vaccinium ohelo, Vaccinium reticulatum, which is basically a type of, it looks pretty much like a cranberry, but it grows in Hawaii. So, we're always looking for new species that we can look into and maybe make crosses. Rhododendrons and azaleas are other species that are closely related, but cranberries they live in a very unique environment in the wild. So you can see here, there's a layer of sphagnum moss, and then there's like plants that are carnivorous plants around it. There you can see a runner here, basically a cranberry. This is Vaccinium oxycoccus, which is not the cultivated cranberry, but these are plants that are adapted to acidic environments, very low nutrient environments. So here are some pitcher plants surrounding some cranberries. And the wild, sometimes cranberries don't produce fruit, and they're more clonal. So cranberries can reproduce clonally by the runners, and then a lot of the plantings tend to be, a lot of the wild stands tend to be clonal because one genotype takes over. And then there's a little bit of fruit you can see on the top of this picture here. I'm gonna see if I can get my cursor on them. You can see there's a fruit there and there. But overall, fruiting is not like, is not present in all of the populations in the wild. So this is one of the things that when we go to wild populations, we look for, is which give fruit and which don't. So again, we have two species. The cultivated species is Vaccinium macrocarpon, is shown in red here, in dark red in lines. Is mostly a species that lives in the eastern United States and the eastern North America, from Minnesota all the way to Massachusetts and north to Canada. And, originally when cranberry was started to get cultivated, people selected native or wild plants from there, and then they planted them. Then Vaccinium oxycoccus is a close relative that sometimes is diploid or tetraploid, and that's more widely distributed across North America. And we're trying to see where we can use it for crosses and things, but this species, the tetraploids don't cross well, and the diploids will cross readily. But one of the issues is the fruit is way smaller, and the plant itself is more like a miniature version of macrocarpon. Phenotypically, the plants can be easily differentiated. One is basically small, and the other one is large. And genetically, you can see that we have used multiple types of markers like SSRs and SNPs, and the differentiation is very clear at the genetic level. Here's a picture. Oxycoccus is in red and macrocarpon is in blue. And then we also have tried to go out all over the country and look for wild plants. We have discovered some populations of macrocarpon in the west and the far west of the United States, which was never reported before. And we're also looking at the genetic diversity because of clonality. Sometimes people used to say that cranberries don't have any diversity, but we've shown that that is not true. There's a lot of diversity. There's a lot of structure because of that clonality. And then we also are looking for populations that produce fruit, which is kind of a rare, it's more of a on the rare side to have fruit. Not all wild populations produce fruit, and not every year particularly. So we've done kind of a survey of the United States and tried to collect materials, and this is how we built our collection. We collaborated with some Native American tribes sometimes in trying to collect these materials. And, of course, Native Americans knew about cranberries even before the settlers moved in and really liked cranberries. So cranberries are a natural food for the Native American people. They used it as a preservative. Because of the high content of acids like benzoic acid, they would mix it with meats and different grains and things that they had, and they created basically what I call the first power bar, if you will. So it's a way to preserve food over the winter. They use it as medicine, as fabric dye, and for them it's a sacred fruit. I've had a chance to have some connections with Native American people in the state here, particularly the Menominee. Just talking to an elder, the fruit, they call it piakamen, and I'm sorry if I'm not pronouncing it correctly, but just listening to him, that's how he pronounced it. And many other tribes in North America knew about the cranberries and the benefits of eating and cranberries and gathering them. And of course, when the settlers came in, they came across the berries, and then they called them cranberries, and then that word became cranberry later on. And of course, in Spanish, we call cranberries arándano rojo, which is if it's blueberry, which is closely related, we will call that arándano azul, so blue and red. But just having a chance to maybe meet with some of this Native American tribes, I've been trying to figure out ways to connect with them and collect fruit in the reservations. And I had a chance to go to the Menominee Reservation and work with the College of the Menominee Nation. And, we helped develop a formal and informal science education curriculum that just basically values and infuses indigenous contributions to scientific knowledge. The unit was a middle school unit that they called All My Relatives, and it just captures the experience of the indigenous people, in this particular case, the Menominee, and to the science learning process. And all this was in the context of the Menominee Forest, which is one of the largest preservations or Native American preservations in Wisconsin and maybe even in the United States. And it was a very nice way to connect. They informed us where there's populations, and our studies and trying to find wild populations. We use some of their ideas and everything. We didn't get to collect in the Menominee preservation yet, and so that's one of the things that I'm working on, trying to see if we can get in there and find maybe some unique genotypes. But again, here's a photo of a cranberry flower. You can see the resemblance with a crane, and that's why it was originally named Cranberry, and then again they just dropped the E and became Cranberry. For production, I have that to say that Wisconsin, of course, is a very important state in production. We produce about 60% of all cranberries in the country. Massachusetts coming second. This is one of the reasons I was hired to be stationed in Wisconsin, because of the strong presence of cranberry growing in the state. We produce of all the acres and all the production, 95% of the cranberries are processed. Only about 5% is consumed fresh, and the main usage is juice and then dried. And dried is becoming like the most important product now, because a lot of people like it, in cereals and in different, products that can be incorporated. So that's why dried is probably the most valuable product that we have in cranberries. And of course, it's somewhat a recent, usage that people have been interested in cranberries. That it has been popular. I guess it has been available for a while, but I myself didn't see it all the time growing up. And I guess people are interested in the phytochemicals and the health benefits that cranberry may bring. They're full of vitamin C, fiber, manganese, pigments, and they're cranberries are linked to many health positive effects. The growth habits is also very interesting in cranberries. Cranberries are long-lived perennial, and they tend to have a biennial bearing, although this biennial bearing is somewhat changing because we're breeding, to not have this so strong, this biennial bearing effect. But here's a picture of a cranberry branch that we call runner. So the lower branch here would be a runner, and then the uprights would be this upright branches coming out of a runner, and the ones that carry fruit that year will be called reproductive uprights, and the other ones will be vegetative uprights. So that's just a little terminology for cranberries. And here's the cycle of cranberries. I mean, just the cycle is somewhat complicated, but just to say that in the winter, cranberries are dormant. In the spring, they wake up and they start growing, and then by June we have flowering, and then around July we have bud set for next year. So if this is a cultivar that is not so much affected by biennial bearing, it might be setting a bud for next year. And then we have fruiting, and then we have maturity, and we have harvest around October. I harvest around September, but a lot of growers harvest in October and maybe even into November. So we just finished harvesting. Here's a flower. Here's the bud that basically starts growing in the spring, and then that produces a flower. Of course, this is a field full of flowers, and then of course we're bee pollinated. Here's an example of some runners. You can see those long branches. They basically allow the cranberry to colonize ground. Cranberries are kind of low to the ground, somewhat low to the ground, and so these runners allow the vine to spread. And here's an upright. So you can still see soil in this planting, but the plant is already producing fruit, which sometimes could be disadvantageous because then those fruit may fall and then seed may germinate and contaminate the planting. So that leads me to propagation. Essentially, a cranberry cultivar is one genotype, and that genotype gets propagated massively, and then acres and acres are planted from that one genotype. So you can do it, commercially, like in this photo, and then take those plugs out to the field, and then they use different equipment, and different techniques to plant. But this is just a tractor that sits a bunch of people in the back, and then the tractor makes the burrows where the people are putting in the plugs. And when the field is finally planted, you can see basically lines of genotype, the same genotype, basically just planted at a certain distance. And then as those plants grow, the runners will basically take over the land and just colonize the whole planting. But essentially, you see the height there might be the height of the planting. I mean, like I would say 1 to 2, maybe, things that are very old can be like 3 feet deep. But in general, we stay around 1 to 2 feet deep. So here's another planting recently planted, and that's already kind of spreading a little bit, but eventually that will become a carpet, and it will be full of cranberry vine. And another way to propagate is mowing. So an established planting will be mowed, and that mold will be collected into bales, and those bales will be just spread over a new field. And then they use different equipment to kind of make contact between the vine, the woody plant material, and the sand, or the whatever soil it is. Then they might even spread soil on top of the vine. So in this case, sanding, they call that sanding. And that will, then they will put water on it, and that will allow the propagule to basically come back and just colonize the new planting. So this is because cranberry plantings are clonal. Then it's very important to have the appropriate material. Licensed propagators are very popular. Less and less people are doing mowing, although mowing is still popular. But I think many people are starting to use more and more propagators. And then also genetic testing for especially if they are doing mowing, they're doing more genetic testing. About the harvest, so here's a planting that looks more mature with the vine already covering the ground. Some of these are plots that are breeding plots, so you can see spaces in between the plots. But when the plants are originally planted, you can still see sand through this, like what you see here. But they start flowering and fruiting somewhat quickly. By the 3rd year, you might already have, or maybe 4th year, you may have fruit already growing. And so you have to manage for fruit so that fruit doesn't land in the fresh sand and then maybe germinate seed. But when the planting is mature, you shouldn't see the ground. And like this one is a little bit more mature planting, ready to harvest. And the way that people have harvested before is just using any labor available. In the old days, a lot of Native American people would work in the cranberry marshes and help harvest, along with women, children, and anybody that was available. Here's a great picture that I love about the harvest in 1880. Just shows families going out there. And this particular photo is more of a wild stand. So people would go to a wild place, kind of clean it up, keep the grasses away, keep some of the trees away, and just kind of clean it up. And again, some of these wild stands, not all flower and fruit. So, so people would go and find the ones that were more productive wild stands and then harvest from those. So here's a little girl carrying a tool. So that would be the kind of tool that would be used for hand harvesting. And at this time in this photo, she was probably doing harvesting in dry cranberries, and she has a little bucket where she's putting the cranberries in. And then later on, they kept using the same kind of tool, but they decided, somebody had the idea that let's put water on the planting because cranberries are tolerant to water, to flooding. So then you can put water on them, and that allows this type of equipment, this type of tool, to be more easily maneuvered in trying to collect the cranberries. You can see that in this picture here. So here's another picture of water-aided harvesting using like hand tool. And of course, that is because, you know, cranberries float when you put water on it. If you put enough water, the cranberries will kind of come to the top. So you can skim any kind of tool, you know, close to the surface of the water, and then that will allow the cranberries to be kind of removed gently without destroying the vine. And so that will protect your harvest for next year. And so, you know, water harvesting really changed the cranberry industry around the 1950s. And that became like the engineering. It's like a feat of engineering, if you will. There's all kinds of ditches, water moving right and left, north and south, and all the water has to come back. And there's not enough water to flood all of the marsh at one point. So, you know, people have to you know, flood it by bed and, you know, using different strategies to do it in sections. And so here is a mechanical harvester in the 1940s. This was a hand pushed more, basically harvester. Here's an example of a tractor in the 1950s when this became more popular. You know, people started actually adapting tractors, and everybody makes their own machine. So here's another guy with a different type of machine, but it's also, you know, type of tractor, gas powered. And here's another one. Here's one last one. And then more recently, you know, we have big companies making big machines to actually make this easier. You know, for some reason, this type of machine that has those basically long needles, if you will, that allows the vine to be moved, but it causes less damage. So that is always one of the things that we're looking during harvest is we don't want to damage the cranberries, but the cranberries are pretty resilient. They can take a big tractor basically running over them, and they're fine. I can walk through them and they can hold me. I'm a big guy and they can hold me okay. After the harvest, basically all of the cranberries are floating around, and they have to be basically corralled, if you will. So we use a type of boom that basically brings the cranberries together. As the cranberries get moved together, they move towards a basically a bridge or, you know, escalator that will bring them over to the truck. And then different cranberry growers use different techniques. Some people use water, some people use an elevator, some people just basically you know, use whatever ingenuity. A lot of these machines are not, you know, commercially available. It's just what people make in their own farm. So there's a lot of diversity on what people are using to do this different tasks. Some people even clean the cranberries before they go to the truck, and some don't. So you can see here, there's a truck where the trash is being removed, and then the cranberries are being dumped in a truck. And of course, people get very you know, fancy with some of their harvest. And so here's just a picture of the state of Wisconsin kind of drawn during the harvest in the cranberry marsh. And here's a picture of our chancellor and our dean, and just enjoying a harvest day in cranberry country. With the domestication, I also have a few stories, but cranberries basically began to be grown more of in a cultivated setting around 1816. Before that, everybody just went out to the field or the wild stand and just collected berries. And the way that these first plantings were made is that people went to a wild place, collected just a sod or a piece of cranberries with roots right from the wild, and then planted them and started basically taking care of them and cultivating them. But the cultivation quickly spread in the US, and by 1853, we were already growing cranberries cultivated in 1853. And then the US Army and a lot of the armies around the world, they started using cranberries as a source of vitamin C. And so that increased the popularity of cranberries and rapidly became an industry. The original plantings were basically selections from the wild. And people just went out and looked for cranberries that looked bigger, that were maybe more juicy. You know, but I think the original selections around the 1830s were probably just related to fruit size, And they were just, and then maybe the ability to, those wild stands that had fruit all the time. As I mentioned before, sometimes wild stands don't have fruit. So here are some of the names of the cultivars that were originally selected from the wild. In all, there was about 132 wild selections documented. And the first breeding program started in 1929. Here's a picture of the Wisconsin station that was part of that program. And that program kind of ended around the 1960s and produced 7 cultivars. And one of those cultivars was Stevens, which is the major cultivar today. About 40% of the industry grows Stevens. But this little figure here on timeline kind of just shows that Cranberry only has had 2 breeding cycles and basically 1 selection cycle from the wild. So between 1835 to 1960, all of the Cranberry cultivars were wild selections. So for 125 years, we didn't have any breeding. And then around 1929 to 1960, there were 7 developed cultivars. And then from 2003 to the present, there's now maybe 10 more cultivars. So we have maybe 17 to 20 bred cultivars, and if you will, 132 wild selections. So here's High Red, which was the first modern cranberry cultivar, or this was developed in 2003 here in Wisconsin, and this is just a comparison between High Red and Stevens, just kind of showing that High Red is, as the name indicates, just more, it accumulates more pigment. And here the cranberry breeders were very few people. My predecessor, Brent McCown and Eric Selden, are shown here on the left. And then you have the Greg Oleski family. Ed and his dad, they have been doing breeding. They have a private company here in Wisconsin, and they're doing an amazing job doing some breeding. Then, of course, Nick Borsa, who has been my mentor for a few years now, and great breeder. And then there's a picture of me there, and the newest breeder, which is my counterpart in New Jersey, is Jeff. And so that's a small group of breeders, but we work together well. And here's a cranberry domestication figure just to show an example of a wild selection there on the top corner of the left side. And you can see this wild plant is just random, but the selections are on the side of it. There's 4 selections there, and just I think the selection was mainly just for fruit size, just try to find something that is productive in the wild all the time and that is a little bigger fruit. And that was pretty much the first selection cycle. And then the 1960s, hybrids were released that basically combined the most productive wild plants and that had bigger fruits, and that gave rise to a little bit more productive plants that had bigger fruits. And then in this current cycle, I think breeders are working on color, they're working on fruit size, we're working on basically just more production and just more appealing looking fruit. Again, things that cranberry breeders are looking for, that we're looking for, is improved color, fruit quality, nutrition, rebudding, which is we're trying to reduce the effect of biennial bearing. Yield is always important. Taste, early maturing, cold tolerance, and pest disease. Although here in Wisconsin, we're doing less of these last 2. In the East Coast, they're doing a little more of these last 2 traits. And just now I'm gonna get into some of the research in my lab, but just to say that I was hired in 2010, at the end of 2010. So in the beginning of 2011 is when I started trying to develop some molecular resources. As the first thing that I tried to do. And I remember going to the gene bank and finding that there were only a few sequences for Vaccinium, and for Cranberry at the time, I think there were only 15 sequences. So I was like trying to develop some markers, and I said, well, I don't have a lot of choices. There were no sequences there that I could use to develop any type of marker. So I was beginning to think what type of marker should I develop. And at the time, next generation sequencing became really possible for crops like cranberry that are somewhat minor crops and, with a little bit less funding. So we did some 454, we did some Illumina. And so based on that, we started really accumulating very quickly a lot of sequence data that we could use. And the first thing we did is we assembled the organelle genomes and published those. And then after that, we started developing some SSR markers just from basically a search that we did with using the 454 sequence data and the Illumina data. And we did kind of an in silico approach to discover SSRs and test them kind of in the computer before we actually started ordering a lot of primers, because we didn't have so much money to start ordering a bunch of primers. And so we developed our first map, in Cranberries around 2013 with 100 markers. Then later, we added markers to this, and it became like almost 600 markers. And all these populations were developed by Nick Borsa at the time. One of the problems that I had is that I had no material, no populations. It takes 3 to 5, 6 years to develop a population, and planting it a number more of years. So I kind of partnered with Nick Borsa and with Jim Polashuk and Rutgers, and we started doing things together. So all these maps are in collaboration with them. Here's a third map basically that we did in 2016. By the way, I'm gonna go back just to show you that this is 2013, this is 2015, this is 2016. And so here we have now access to development of SNPs markers that we did by the genotype and by sequencing approach. In fact, we developed these markers with the use of Cornell at the time. And we had a kind of a draft, a very low, I would say, lower quality draft of the Cranberry genome. And based on that, we were able to use this GBS technique and just, get our SNPs and add that to the SSRs that we had. And then later, we added other populations. So we had, in this case, 3 populations. And these are populations that we're trying to study traits also. But, and we were hoping to do QTL markers analysis with them. But we were able to develop a composite map out of 3 populations that we're actually studying for traits also. So here, this is in 2017. And this is kind of where we are right now, trying to use these maps and this technology that we've been developing now to actually add traits and just really start understanding the genetic structure of the Cranberry genome in terms of traits and everything else. So all the things that we have been able to do thanks to this technology is to compare ourselves and use information from other crops. So we've done some comparative genomics with kiwi fruit, with grape. We haven't really used a lot of this information, for anything, just to say, well, where we are in terms of other crops. But we're working more closely with blueberry. Blueberry is very close to us. I will show you some pictures of that in a minute here. But just going back to a little bit of the work that we did with SSRs is that, we did develop about 700 SSRs that have been very useful in my program. And, so here's just a little bit of what we do with them. We are very interested in finding out all of these wild plants that were originally selected. So what are they? What is their genotype? And how are they used in crosses that produce the first cycle and the second cycle of cranberry breeding? A lot of these things are noted in different books and historical material, but we wanted to corroborate some of these things. So here's an example of a cross that is very important. And so Stevens was crossed with Ben Lear. Ben Lear is a wild selection, and Stevens is a first generation hybrid, and that got crossed to high red. So we actually can show that is true. In some cases, we've discovered that the crosses that the breeders made were not exactly the way they intended or they thought, and so we corrected that where we can. And we're just doing a database of basically the genotypes and parentage. And I have a new paper that is going to come out with the genotype of, like, I believe it's over 20 cultivars that are interesting and useful in cranberry breeding, and then again, we can differentiate with simple SSRs. We can differentiate and identify cultivars, and that's one of the things that we concentrated a lot, is to try to preserve a lot of these materials and go find them and put them in collections, and just make sure that people are growing what they think they're growing. And then off of this is also to help growers test their fields, again, since everything is supposed to be a clonal planting. In this particular case, this is a marsh where everything was supposed to be Stevens, and then the grower was having some problems in some beds, and they told me, why don't we just do a like marsh wide study where we collect samples and then figure out if there are any differences in bed genetic composition. And so we did find some of the beds that the grower was having problems, like this one here on the top. You can see all the yellow color dots are basically not the correct genotype, and all of the red is supposed to be the Stevens genotype, which is the one that they were intended to grow. So we do this all the time. We've tested over 500 growers samples here in our state. We test 20 to 30 to 40 grower samples every year. I mean individual growers that send us samples, and we do anywhere from 24 to 100 samples per grower, depending on what the problem they're having, and we try to help them clean their operation, if you will. Some make decisions on renovation, and some make decisions on cultural practices. And then again, the SSRs have been useful to us to study vaccinium, in particular crosses with the closest relative of cranberries, Vaccinium oxycoccus. Then we've been trying to cross with lingonberry, with corymbosum, and different species. We do have some crosses between the cranberries, and so our initial efforts have been applying SSRs, and now we're applying SNPs, and we're actually trying to map some of these crosses, segregate widely, and it's very interesting just to see the segregation on some of these crosses, because the plants are so different. And then one thing that was very encouraging, because in the beginning I wasn't sure if I should pursue some of the blueberry cranberry crossing breeding, but then we used the SSRs, and we demonstrated that cranberry and blueberry have basically a lot of synteny. The chromosomes align perfectly. There's no major translocations or mutations that we can find. And more recently, in 2021, we finished our cranberry genome. This is again, so just a little bit ago, the Stevens cranberry genome using PacBio. We had the 12 chromosomes, and most of the genome was assembled in there. We still have a few gaps. We're still looking to develop the genome, and we also developed a genome for the closest wild relative of cranberry, a diploid, basically accession from the oxycoccus that we call microcarpon. And we're hoping now to get together with Backcap and with Bringing Insight and different people that will help us create more genomes. We have plans to create a cranberry pan genome based on 10 different genomes. So that's going to be coming up pretty soon here. And also a genotyping platform that is going to have 21,000 genes or 20,000 genes, plus another platform that is going to be developed with Bringing Insight that has 3,000. And now we're hoping to really be able to have really deep coverage and excellent marker density for any study that we want to do. Again, and based on this genome, we also were able to show again that the blueberry genome and the cranberry genome are very, very synthetic. So there's here you can see the chromosomes of cranberry in different colors, and we're showing the 12, and then the tetraploid blueberry chromosomes. You can kind of see the color that corresponds to what cranberry chromosome. So that's very encouraging, and that kind of supports some of the research that I've been doing in terms of breeding. In terms of phenotyping trait data, we've been trying to follow the traditional measurements and add measurements. So when I came here, people told me what traits I should measure. And I've been doing that plus creating new traits. And part of the work that we're doing in terms of trait measurement is related to the backcap project. Massimo Yorizzo in North Carolina is our team, basically our PI, our leader. And of course, I work with Borsa and Polashok and Amaya Tucha and many different people on this project. But we're looking for traits that are important for fruit, for dried fruit. So, things like color, shape, fleshiness, and firmness are really the most important traits for this type of product. We have collected over 70 fruit quality traits. Some of them are related to external appearance, internal structure, and firmness, which we can measure many different ways. But just to show the cranberry external appearance, the way that we do it is in around 2016, we started developing this basically way to just take a picture and see if we could just, look at this basically from a picture standpoint, organize the berries, put a little, basically a standard on the sides, and then basically take a picture with a very rudimentary little picture stand, if you will, that we came up with. One of my students had a little bit of engineering background, so he built this from our lab, from just pieces of scrap, wood that we had and everything. And so, we started like, making a little R code that would recognize the fruits and compare them to a standard, and then basically very easily tell simple things like the perimeter, the length, the width. And then another student came around and said, well, maybe we can use the pixels to like look at how dark the fruit is, whether it's light or dark. And so then we did kind of a fruit color analysis. We published this, I believe, maybe in 2017 or, around that time. And, essentially it's an R code that just, looks at the photos and then spits out an Excel sheet with just different ways of looking at the fruit. And, all of these measurements are related to fruit shape, size, and color. And color variation, I think, is one of the most interesting ones, because in cranberries, we want to have all the fruit to kind of be color and not basically blushy. So internal structure, we kind of took the same approach. We were doing a kind of an outreach event where students, little kids, would come over and cut cranberries and then use the cranberry to use as a stamp. And it was like a very colorful experiment. We would just like use different colors, and then one day we said, why don't we use this to actually see if we can measure the internal structure of cranberry. We noticed that that was back in 2011 when we started doing that outreach event. And then we said, we noticed that there was very different inside of the cranberries. And so we started figuring out how to measure these stamps. And we created another little software that measures the cranberries, and it kind of creates a way of kind of rotating the image and measuring the inside of the cranberry, creating about 10 parameters related to the flesh inside or the absence of flesh inside of the cranberry. And we're using that now to analyze the fruit in the cranberry backcap project. For fruit firmness, we've tried many different things. Among them, the double compression, which tends to be the best test for us. Using different strains, different speeds, double and single, different things, but the main idea is that when you press a cranberry, there's a force required to press that cranberry a certain percentage of its size. And so based on that, we're developing different ways of looking at the data. You can do a first compression and a second compression and compare the peaks and many different things that they're somewhat, frankly, out of my expertise, but we're consulting with food science people that are helping us decide what are the best techniques. Other things that we've tried is puncture, and then the blade, and then the Kramer test, and we're working with all of those traits and see which ones can help us determine the firmness. We'll also are moving towards more of a sharp knife, and those are the work that we're going to do later. But in general, just to say that cranberries can be differentiated, different genotypes with different properties that growers tell us this is firmer, this is not so firm. We can differentiate them using these firmness techniques. And we also store the fruit and then we pull it out, and then we can see the decay of the fruit using these techniques. So we're happy with what we're getting. We're trying to use this data for further QTL mapping and other studies. In terms of yield, we are collecting yield data in terms of like upright yield. So you can see, you can take an upright and count how many fruit are there and the weight. And then we're also using the plot yield method. So basically we have a square and we collect all the fruit in that square. One of the problems with this method is that sometimes you can get different results. The error can be high sometimes. So we're trying to figure out how to change, how to look at the plot data. I think this is one of the biggest challenges in Cranberry trait data, is the collection of yield. So we've experimented with images, we're experimenting with drones, with sensor technology, and the idea is like maybe one day, this is Luis Diaz Garcia, one of my students that some of you may know, and we were always thinking about how to use different technologies so that we can be drinking coffee while we collect data, but you know, we're somewhat far away from that yet. There's some advances. We used photos, you know, microwaves. I'm sorry, is that a question?
We've got only got about 2 or 3 minutes left. Did you want to do some questions, or you keep going either way?
I have like maybe a few more slides left, but you know, we can leave it here if you want. You know, basically trait data. I was going to cover something on QTL mapping. You know, we have a ton of QTLs developed, maps, some candidate genes, you know, ton of data that we have collected in populations, breeding, you know, a little bit on the phases of breeding. Let's see. Let me get to the last part here. I just want to show you one more picture. A little bit on the, you know, harvest, field plantings, and one of the most exciting things is our blueberry crosses. So we have some hybrids with blueberry, and we have some fruit that is coming out of the blueberry cranberry hybrid. So with that, you know, I can stop and just take some questions if you, I apologize. I thought I had a little bit more time.
Okay, we've got one question from the chat from
Yes.
Justine. Can you see that? Can you see the chat, Juan?
Yes.
Okay, great.
So is there any path towards improving fruit set through breeding? 5 to 7 flowers form only 1 to 2 fruit, or at least, is in the case of the Massachusetts box. How can that be improved? Yeah, so that's one of the things that I was showing there. I mean, that's why we're collecting fruit upright data. So, you know, I'm collecting not only the overall square footage, you know, yield, but we're also looking at the upright level. And so we're counting things like how many aborted fruit, how many, you know, how many flowers, and then what was the fate of those flowers throughout the whole cycle. And so we're really looking at that. And you know, I think that there's something to that. There are genotypes that have more energy to give to fruit, and that's actually probably one of the ways that cranberry breeding is going that way right now. Is just we're looking at uprights themselves as the unit of selection.
Okay, I'm afraid we're about out of time. Dr. Zalapa, I really appreciate your time here, and maybe we could leave your email address in the chat or something for folks in case they want to talk to you about anything else. Would that be okay?
Yes.
Great. Okay. Well, thank you very much.
Thank you.
This has been a production of Cornell University, on the web at cornell.edu.